production of mushrooms. Mushrooms, basically, they are fungi, belongs to the class Basidiomycetes. These are the large fruiting bodies, consist of caps, gills, and style. All mushrooms are not edible. Few mushrooms are lethal to human beings, as they are responsible for production of toxins. Nutritionally, mushrooms are very important. They are low in calories, fat, and sodium with no cholesterol. They have high fiber content and also contain several vitamins such as vitamin C, vitamin D, pyridoxine, and niacin. It also supplies essential elements such as copper, selenium, potassium, iron, and other important minerals such as phosphorus, zinc, and magnesium. It has high protein content, about 20 to 30% by dry weight, and it also shows medicinal properties as building up of resistance power of humans. Edible mushrooms are grown in several parts of the world. For example, Agaricus bisporus and Agaricus bitorqueus, which is generally grown in western regions where the temperature is approximately 15 to 20 degrees Celsius. This is a picture of Agaricus bisporus. It is also called as button mushroom because of its shape, and generally it is cultivated on compost. Another example is Lentis edodus, which is grown in Japan, popularly called as shiitake mushroom. The cultivation method is very unique. It is cultivated on hardwood logs as shown in picture. After 8 months, logs move to the site optimum for the fruiting body formation. Harvesting of cap, it is done for approximately 6 years, and it is a low cost process. That is a major advantage, which can be grown under natural environmental conditions. Fruiting bodies form in spring and fall at temperature of 12 to 20 degrees Celsius. Another mushroom is oyster mushroom. This is also named because of its shape. Scientific name is Pleurotus austriatus. Oyster shape of fruiting body as shown in picture, and it is generally cultivated on plant residues and industrial waste. Another example is paddy straw mushroom, that is Wolveria wolvesea. It is a tropical mushroom as shown in the picture. It is suited to high temperature of tropical climates. Traditionally, the method of production utilizes rice straw as the substrate with minimal composting before inoculation. Different mushrooms require different production conditions. Fruiting body formation is often possible for temperature range as up to 10 degrees Celsius and in a moist atmosphere. The mushroom mycelium usually exhibits some enzyme activity against cellulose and lignin and can assimilate a wide variety of crude carbon sources. Once established, the mushroom mycelium produces antibacterial and antimycotic substances which protect it from the microbial attack. The production of mushroom or mushroom fermentation, there are 4 steps. The first step is preparation of substrate on which the mushrooms are grown. There are several substrates, suitable substrates which are available. For example, manure, corn cobs, straw, and other agriculture residues. Although municipal garbage is also used. For indoor cultivation, there can be use of cereal straws such as wheat, rye, rice, and oat, which are easily available and comparatively they are very cheap. Other substrates are wood logs from elm, beech, alder, ash, and cottonwood. Thicker hardwoods such as oak. Enriched sawdust can be also an effective substrate. Few other substrates are compost, paper, and paper products, cardboard, used coffee grounds, used tea leaves, gardening debris, and other organic materials such as seed shells and banana fronds. Second step is production of spawn. Spawns is the inoculum, which is basically used for the propagation of mushrooms. This inoculum consists of mycelium or primary filamentous growth of mushroom fungus. The inoculum is prepared or the spawn is prepared in following stages. In the first step, mushroom spores are collected from mushroom cap. Once after collection, the spores can be preserved for indefinite period under refrigeration conditions due to their high stability. The mushroom spores are then transferred onto potato dextrose agar or malt extract agar plate and incubated for a week at 25 degrees Celsius for the cultivation or the growth. Then the spores born on these plates are transferred into 50 ml of liquid medium containing rye, millet or wheat grain medium with 49% whey, 50% water and 1% calcium carbonate. Calcium carbonate is necessary for the neutralization of organic acid which may be produced during the incubation period. Rye acts as a support for the mushroom growth. After 2 weeks of incubation at 25 degrees Celsius, the contents are again transferred to 300 ml of flask of same medium and incubated under similar condition. These transfers are repeated several times using 2 liter thereafter to 1 gallon. So this is how the inoculum is allowed to build up for the production of spawn. The mycelium inoculum, it is now called as spawn and it is used to inoculate the substrate. This is the picture of spawn, which means these are the grains of rye, wheat or sorghum which are coated with primary mycelia. Then the next step is inoculation of the substrate. As it is mentioned earlier that there are several substrates that can be used for cultivation of mushrooms. So based on the type of substrate, the method of inoculation may change. Say for example, in case of compost, which is used as a substrate for the production of button mushroom, that is agaricus, the spawn is added to each tray containing compost. The trays are then placed in an incubation room which is called a spawning room at temperature of 17 degrees Celsius and a bed temperature of 21 to 29 degrees Celsius for approximately 18 days. Bed means it is a bed of compost in which the spawns are inoculated. Next step is the formation of fruiting body, that is formation of mushroom. In case of agaricus mushroom, after inoculation of a bed, the mycelial growth goes through a lag phase for several days. By the end of first week, there is sufficient growth to produce large amounts of CO2 and heat. At this level, airflow is required for the last 11 days of 18 days of spawn growth phase. Now the bed is ready to support the healthy production of mushrooms. Then there is a step called as casing. It involves the addition of a 2-inch layer of topsoil to the top of the bed. The casing is moisturized to about 50% saturation to allow the even spreading over the surface of the trays. Then the trays are shifted to the production room where the temperature is maintained between 14 to 17 degrees Celsius. The first step in the formation of a mushroom cap is mycelial cell differentiation to the growth of fruiting body cells. The fruiting body starts as a pinhead sized mass of mushroom tissue at or near the surface of bed. The effect of carbon dioxide is found to be quite important in the formation of fruiting bodies. Usually the CO2 concentration in the production room is raised to 0.5% in order to suppress the formation of fruiting bodies for the first week until the mycelium is completely penetrating the casing. Other factors which influence the fruiting body formation is the air temperature and the nature of casing. For example, at a temperature about 22 degrees Celsius, Agaricus bisporus stops formation of fruiting bodies. Some varieties of Agaricus bitorqueus will, however, form fruiting bodies at higher temperature. So this factor is variable. After 3 weeks of in the production room, the mushrooms are ready to be picked up from the trays. In case of Agaricus bisporus, mushrooms are all mushrooms are all hand picked and trimmed. At the end of a 9 week period in the production room, the trays are removed and steamed to kill mushroom viruses, any insects, nematodes, contaminating fungi, or the weed molds which may have entered the bed. Finally, the mushrooms are ready or they are harvested. This is a picture of oyster mushroom getting being harvested from the beds of rice or wheat straw, and the harvesting of agaricus from compost, and finally they are packed and marketed.